Good day everyone. Today we'll talk about the DNA extraction from human cheek cells. The group members of Group 1, which is Chinchontic, Kliman, Frias, Gayosa, Lagudas, Lontok, and Morales. Now, moving to the introduction. DNA, often referred to as the blueprint of life, contains the genetic instructions that determine our physical characteristics and biological functions. This remarkable molecule is present in every cell of our body, holding the keys to our genetic heritage. It plays a critical role in influencing how we grow, develop, and function. By studying DNA, we gain insights into the science of inheritance, evolution, and how traits are passed down from one generation to the next. The process of DNA extraction is essential for this understanding. It involves isolating DNA from cells, tissues, or organisms for analysis. This step-by-step process typically includes: number 1, cell lysis. It is breaking open the cells to release DNA. Number 2, DNA purification. Removing proteins from cellular components. Number 3, DNA precipitation, where DNA is separated from the solution. Number 4, and the last one, washing and resuspension, ensuring a clean and a usable DNA sample. This procedure is fundamental in molecular biology and serves as a gateway to studying genetics in greater depth. Let's now proceed to the next part.
So for our methodology, to extract DNA from cheek cells, first to bring the materials needed: saline solution on salt water concentration, dishwashing liquid, ice bath or chilled ethanol alcohol, paper cups, sticks, and clean test tubes. The first thing to do is make sure that each member brushes their teeth well, then prepare the alcohol in an ice bath. After that, in a cup, make a saline solution using 25 milliliters of distilled water in a cup. Add 2 tablespoons of salt, then stir the solution for around 1 minute. Once it is dissolved, gargle the solution for about 30 seconds. Bite your cheeks in order to destroy some of the cell walls to extract more DNA. After gargling, pour the solution back into the cup. Add 2 drops of dishwashing liquid. Next is to stir the mixture. Make sure that the mixture doesn't bubble. After mixing, get a clean test tube on a 45-degree angle. Then pour the cold alcohol and pour it about half the test tube. Then afterwards, slowly pour the mixture on a 45-degree angle. Make sure that it doesn't mix with the alcohol. Once both mixture and cold alcohol are on the test tube, let it sit for about 5 minutes. Observing the test tube, there should be white stringy clumps forming at the interface between alcohol and the mixture. Get a stick to take a small sample of the DNA extract, then this represents the extracted DNA.
The DNA extraction experiment. Students can expect to see stringy clumps of DNA after allowing the mixture to sit about 5 to 10 minutes. This process is fascinating because it enables them to observe their own genetic material, making the concept of DNA more tangible and relatable. The result is a visible layer of white stringy material that represents the DNA extracted from the cheek cells.
As a result of our experiment, we have demonstrated a distinct separation in all test tubes between the alcohol layer and the DNA extraction solution. Successfully, visible DNA fibers were observed, particularly in the test tubes of Lagudas and Kliman, where whitish, stringy formations were seen at the alcohol interface. However, the other groupmates' test tubes exhibited either minimal or no discernible, means there had less or no visible DNA fibers. But overall, our group quite effectively demonstrated successful extraction of DNA.
In summary, the experiment successfully demonstrated the process of extracting DNA from cheek cells. we use 25 milliliters of distilled water and 1/4 teaspoon of salt to create a saline solution, which is gargled for 30 seconds to collect cheek cells while biting the cheeks to release more cells. The solution was then returned to the cup, and 2 drops of dishwashing liquid were added to break down the cell membranes, mixing gently to avoid bubbles. The mixture was then transferred to a test tube, and chilled 90% ethanol or alcohol was poured at a 45-degree angle to form a separate layer. After letting the test tube sit for 5 to 10 minutes, white stingray clumps of DNA were observed at the interface of the alcohol and solution layers in some test tubes, particularly those belonging to Lagudas and Kleeman. However, some test tubes showed minimal or no DNA, likely due to variations in technique or cell collection.
As for the recommendations, first would be the oral hygiene. Rinse your mouth with water after brushing to remove toothpaste and debris. And 2 would be the saline solution. Dissolve salt in warm water before cooling to avoid undissolved sediments. So 3 would be the gargle technique. So swish vigorously and scrape your cheeks with your tongue for 30 seconds. And the 4th one will be the dishwashing liquid. So use 2 drops of dishwashing liquid for cell lysis. And invert the tube for 8 to 10 times slowly to avoid bubbles. So as for the 5th, which is the alcohol addition, so add alcohol dropwise to form a distinct layer, and use a 1 to 2 ml of chilled alcohol for better precipitation. And 6 would be the observation, so DNA appears white and thread-like in the alcohol layer. Then optionally add a drop of food coloring for visibility. So that would be all for the recommendations, and thank you.